slide we left off with, right? Showing, we had already discussed them in the previous page, I guess page 5. And so now we have a couple of PRS over the cells, okay? Here we go.
Oh, so it's multiple ones. So yeah, it's gastro, right? It could be one. But what could the other one be?
What are you guys thinking?
1, 2, and 5.
1, 2, and 5?
I think it's one of them.
I'm not sure if one. I was thinking 2 and 5, but I wasn't sure about 1.
Is there another one?
Okay, so I'm going to speed along and give you the most time to review. Here's the timer.
Should we do 1? I'm just gonna stick with those. Yeah.
Do you know how many there are? Because one is actually inhibited.
Oh, baby, we got the right. Oh.
You guys cheated, just looked and said, oh, there's 3. 2 have an up arrow, 1 has a down arrow. All right. But this brings, oh, no, I promised 2. Here's the second one. But very good, you at least got 2 of them.
I was sympathetic.
Are you saying sympathetic or
3 and 2. I think it's sympathetic. Acetylcholine's gonna make your muscles
inhibits
go more, right?
digestive
So,
what?
I mean, basically.
I thought something was first.
No, I have.
Yeah, ball. Great. So it'll be a little dimmer. Yeah. The premier lecture hall at UC. Here we go. All right. Timer. All right. So actually, all of them secrete acetylcholine. I can't believe nobody questioned me on this. Because in the autonomic nervous system, the preganglionic always secretes acetylcholine, whether it's sympathetic or parasympathetic. But you guys figured it out. I was asking for, isn't that what you guys went through in your mind? I know he wants the postganglionic. Yeah. The somatic also secretes acetylcholine. Neuromuscular junction, but I asked the autonomic nervous system. Okay. All right, so here we go. Very quickly, I've taken these notes and separated them into 3 parts to help you out. The cephalic phase occurs all in your head. Your sight, smell, taste, those are all your special senses in your head, and then of course using your brain thought of food. Actually begins things in your stomach. So that begins with, what do the endocrine cells secrete? Gastrin and histamine, like we just learned in the PRS, but somatostatin is inhibited. Somatostatin inhibits acid release, so it does the opposite of what gastrin and histamine do. Then what do the parietal cells release? First of all, the vagus nerve releases acetylcholine, which has a direct effect on the parietal cells, causing them to secrete hydrogen ion. With that hydrogen, potassium proton pump. Lots of ATPs needed to do this. Okay? And I apologize. Take a deep breath, and here we go to the next slide. You guys have no idea how fast I went in the 11 a.m. They were like in shock. They just became numb. Hoping that it was going to be recorded, and of course, it trying. I'd say thank you, Jeff. Do you think it's good? Okay. All right, but he's working on it. Here we go. All right, next is called the gastric phase, because now it's not just the thought, smell, and all that. Food enters the stomach. Then it's the gastric phase. What starts it is distension of the stomach wall. That will then stimulate the enteric nervous system, which is the gut brain, the local reflexes that can do on their own without other nervous stimulation. But also, the vagus nerves will also, like we just learned from the previous one, they go to parietal cells where their stimulation does for more hydrogen ion. Same to enteroendocrine cells that are stimulated, and the somatostatin continues to be inhibited. All right. Now, in the gastric phase, we have a positive feedback loop again. Parietal cells continuously are stimulated to do HCl, and what finally shuts it off is when we finally reach the gastric juice pH, which is 2, as you guys know. Then that starts off. Look at all the hydrogen ion theories. Okay, guess where the chyme enters next, and what the phase is called? Intestinal phase. And the intestinal phase is stimulated by the partially digested proteins. And we know they were partially digested because of the pepsin in the stomach. But what we're talking about here is acid secretion. So, now, 2 new hormones come into play. Secretin and gastric inhibitory peptide, or GIP. And those come from the duodenum, but they're hormones, so they have to go into the what? Bloodstream, and then have their effect on their target organ. They are not released like enzymes into the lumen. They do the opposite. They'll decrease vagal activity, reduce acid secretion. And the parietal cells, as you can see. Now, if you look closely at the figure in your book, if anybody's looking at this, she made another mistake. But Zach, who was I asked him, how can you Photoshop things in like a second? And he does. I tell him what I want, and you could never know. He was going to be a graphic designer before he decided he wanted to go to med school. As you see, this arrow should be a down arrow. In your book, it's another up arrow. It doesn't make any sense. So we're going to decrease hydrogen ion release. All right, PRS, here you go.
But we're recording and on her iPad. And then if the recording stops working, then we'll just take pictures of the slides.
Yeah, I was thinking about taking pictures and using it. Well, I'm kind of like looking at it to make sure, like, I don't really want it to get stuck. Like, I don't know if it's going to go straight for it.
How much battery do you have?
I think 80, I think, or something. It should be. Yeah, does anyone else have their iPad out?
I don't have mine with me. I used to paper test. Chick-fil-A cups always look like smoothie cups, and it makes me want smoothie cups. Right, and Jordan. Did you have Chick-fil-A for lunch?
Yeah, I just gotta get some.
I got it for dinner. Good.
You're working out,
Yeah, but then I cancel out the working out.
Yeah. Maybe just don't get like something like a
You go to Chick-fil-A, you have a salad.
All right. Okay, let's talk about GERD. GERD They are treated by three things: antacids, which are the cheapest thing, like Tums; H2 blockers, which was all around when I had mine, and I took Tagamet, and there's also Zantac, but they're over the counter now. Tells you how long they've been around. And then the new ones, the proton pump inhibitors, which are the most potent. They block that hydrogen potassium pump, not enabling your acid level to come down, like Nexium, Protonix, and Prevacid. Okay, PRS. And I really apologize, but you'll see how long this whole thing takes.
What are you thinking? Would not make
What are you guys thinking?
Lord. I'm thinking two. Yeah, I'm thinking it's two. Three, he just said, yeah, he just said dessert. Yeah, so it's not 3 or 4. And heartburn is like when your toxin probably messes up. Because it's like the acid is above. Yeah, that's what I'm thinking. Because of the obesity and stuff. Heartburn has to do with like acid reflux.
So to answer this question, you have to know what the LES meant. What is it, Mackenzie? That's right. Lower esophageal sphincter. Okay, one of them's not from that, but the other three are. Do you guys know the big word for heartburn? Fibrosis. Means a condition of fire. What's your hiatus? Anybody remember what your hiatus is? I taught you that last class. Anybody know? Not one person wants to guess. They finally figured out in 11 a.m. It's not dealing with the stomach. It's not dealing with the esophagus. Eric's right. It's dealing with the hiatus is the opening in the diaphragm to allow what to get into the abdominal pelvic cavity? The esophagus. So the esophagus has to get through a hole. What do you think a hernia is? It's a defect in that hole, and guess what my patients had with this? They had their stomach in their chest cavity. Can you imagine? That's going to hurt. You're going to get a lot of reflux. So that's a hiatal hernia. All right, what are the three actions of motility of the stomach? We first have to receive the bolus. Look what your stomach has a volume of, 50 mLs. Look what it can expand to, 1,500 mLs. Did you know your stomach went that small? No. Don't tell me you did, because I can't imagine. But expand to 1,500. What happens is the LES, which is also called the gastroesophageal sphincter, and the smooth muscle in the stomach have to relax to receive, which is receptive, the bolus. The body then relaxes, and you're able to fill. All of this is involuntary, of course, controlled by your brainstem, medulla, and the vagus nerve. Then we got churn. So the churn, we're going to close both of the sphincters, the LES and the pyloric sphincter. The smooth muscles, on their own, due to here it's called the gastric pacemaker, that's part of the enteric nervous system, which is a local nervous system specific for the gut. Also, serotonin will be secreted, and we know serotonin is a constrictor, and it's going to increase motility. And the duodenum forms a form of gastrin different from the one in the stomach called intestinal gastrin. That will stimulate motility. Then we got to empty. The emptying function is going to be the movement of chyme into the duodenum. What does that is stretch the pH finally getting down to 2, high lipid, because you're going to be, do you guys know that mechanical digestion, not just chemical, mechanical is a big part of us breaking down our fats into smaller things. Then the vagus nerve and hormones, and this time 3, secretin, cholecystokinin, and GIP, and then bicarbonate that has been secreted into the duodenum. All of those are at a significant level that the stomach will then empty. It's like it's signed for that. The bicarbonate is going to do what? Neutralize the chyme. And it's from the duodenum. And I'll get into that when we get to the duodenum and pancreas. All right, here's churning. Remember, churning is the function of how many smooth muscle layers in the stomach? 3. 1 to squeeze, or push down, 1 to squeeze, and 1 to twist. So all 3 of those is churning, and both of the sphincters are closed during churning. All right, put away your notes. We're going to have a quiz on your own, and then we'll continue the lecture. All right, here we go. First question. Very good. Paige, will you take my notes out from my notes for just a second? Finish the quiz. All right, timer. All right, it went up. Have you seen this question? I thought I'd try it again. It's still not right. Even if you've seen it. And timer. All right, the parasympathetic is stimulatory. The enteric nervous system is autorhythmic, and it'll do it on its own. The sympathetic is inhibitory, but they all control, okay? So good for the 30%. Everybody understand that now? You need all 3 systems to control. Okay. Don't yell at me. You're right, I'm wrong. Okay. When I find that out in one class, I can't change it for the next one. But you're right, because the soft palate is not elevated and the epiglottis is not down, it is buccal and voluntary, okay? All right. Or lower esophageal sphincter. Yeah, LES. All right, timer. All right. Very good. Remember, ACL has an L in it, parietal has an L in it. Okay, are you gonna do this?
I know, like I got like everything right in that class.
So
Like,
it still appears that it's not being recorded. So we're gonna go. And you guys see we have 5 pages to finish. Here we go. All right, small intestine has 3 sections. The duodenum, the jejunum, and the ileum. You guys already know this from lab, so I'm going to spend 2 seconds on it, but I apologize. It's about 10 inches long. It is retroperitoneal. It has the major duodenal papilla, which is the last structure where the hepatopancreatic ampulla and sphincter come to. Then the papilla is the whole. They have Brunner's glands because now they produce an alkaline mucus. Why? To neutralize the highly acidic cum, so we don't destroy our insides. Then the jejunum is the middle section. It is intraperitoneal. And this is the most active site of chemical digestion and absorption. It's not the longest section, that's the ileum, it's a little bit longer. Ends with the ileocecal valve, and that controls movement, making sure everything is digested before it moves in. All right, so here are the 3 sections, and here's the PRS. You guys, I swear, we gotta go this speed, or you're gonna have a lot to do on your own. The audio should record, but unfortunately, it's the video that you guys would rather have. The one in that
what is it? It's all the above. Like it has to be more than one because the structure is.
But it has to say structures because it's which of the following structures. You know what I mean? You can't say which of the following structure.
If you were paying attention, the next bullet says, describe the 3 progressively smaller folds that increase surface area for, anybody see that? Of one. Yay! So you get the quick points. Yes, all 3. Okay, the first ones are called circular folds. They're the largest, and they are due to ridges involving the the mucosa and submucosa. Then the villus, which you guys saw, villus singular, villi plural, which you guys saw, has the blood capillaries and the lacteal in the middle of it. What's the lacteal for? Fat absorption. Yep, it's part of the lymphatic system. And then, just like the gastric pits, we also have intestinal crypts, which allow more of the chemical digestion to occur, lying right next to the lining. Okay. Both types of cells. And then last but not least, what's found in all of the absorptive cells of the villi are microvilli, increasing the surface area to what, just a ridiculous amount. To the point that we absorb pretty much, unless you're sick, every single nutrient that's possible in what you eat. That's why humans gain weight when they take in more than they need. You will continue to do that no matter what, it won't stop. Found there are brush border enzymes. They usually break down the smaller compounds like disaccharides and small peptides. Okay, I gotta move on. Here's the folds in the figure. Here's the villi. You've already seen this in lab. Here are the microvilli off of each of those cells. All right, what do you think? Here's the PRS.
Yeah.
All right, is this going to be 100%? Ooh, wow, you work with lactose intolerance. You guys are special. All right, we got it. Yay, let's move on. All right. Most definitely of the small intestine. All right. There's two phases, during fasting and during eating. During fasting, the migrating motor complex occurs that even though it's not being stimulated because you're eating, we still have to empty it. It's 21 feet long. Well, about 20, because with duodenum it's about 10. Clears any remaining material, getting it to the ileocecal valve. Good old enteric nervous system, the gut brain. I call it the gut brain because the gut has its own brain. It can depolarize on its own and contract on its own. Also with the assistance of another duodenal hormone called motility. Then during eating, two movements occur to bring it after it's kind of gone from the stomach, and that is peristalsis and segmentation, and this is nervous stimulation by the vagus nerve. Peristalsis, alternating circular, longitudinal, allowing a rhythmic movement of the food through the intestine. But then in the other thing, we got to mechanically digest it and mix it. And that term is segmentation. But this occurs only with the circular layer of smooth muscle. Okay, got to move on. Here is a figure showing the two constricting muscle layers, the longitudinal and the circular, and here only the circular. All right, that table was in your book so that you can see all the hormones. If these hormones are getting confusing to you, I highly recommend you making note cards for these. I told you in medical school I only made note cards for two things: muscles and hormones. Other than that, I didn't really find it helpful. But those two things, they just all sounded the same. Origins, insertions, and actions. All right, now to the large intestine. And please do not think that because I have to skip through these things quickly, that it's not on your exam. It still is, okay? All right,
Yeah.
we're going first.
You don't know what it is. Kind of long.
Of course. No one picked anus, but somebody picked rectum. So they poop out there. Okay. Whatever. All right, 5 main functions. I've given you help to start off with. Passageway for feces is all you got to write for the first one. Absorption of water and electrolytes. The second one. Get the key words down if you can't get it all. Why do we need to monitor water and electrolyte? We got to monitor our fluid and pH. Then secretion of mucus. Why do we need mucus in our large intestine? I'm sorry? Facilitate motility, but more importantly, well, a lubricant, but it actually binds the fecal material together. Can you imagine if it didn't every time you had a bowel movement? You'd look in there going, ew, it's just a mess. No, it's a nice, beautiful shape, isn't it? Well, compared to what it could be, come on. All right, propulsion by smooth muscle, and of course, specifically the sigmoid colon and rectum defecation. All right, what are the regions? I'm not spending any time on this because I've already went over this. It's the pathway. Make sure you know what comes first, what comes second. Make sure you include the flexures. All right, this makes sense, I hope. We got to move on. So I can spend time on the more critical things that you haven't learned in this class. All right, there's your large intestine. This figure, of course, will not be on your exam. Why? It was on the practical, but you still have to know the material and the thought of the passage. And there's the anal canal at the end. All right, let's fill in some blanks.
What?
Pull that down at the top just a little bit, Julia. There we go. No, a little bit more. Wait. Never mind, where am I? Oh, there it is, B, gross anatomy. So, epithelium histology consists of what kinds of epithelium, everybody? Because we're still in the GI tract, it's simple columnar. That lacks what? What is missing off of these cells that's found all the other way? Not cilia. Microvilli, yes. They don't need microvilli because only water and electrolytes, which don't need that, get absorbed. But they have what kind of cells? Goblet cells, right? That secrete what? Mucus. There we go. All right, then we have 3 structures. The T. coli, which give their bands of smooth muscle that shorten the length of the large intestine, forming pouches called haustra, and that can increase the surface area. And then they have special little structures called epiploic appendages. Those are part of the serosa and their fat film. All right, what are 4 functions? Did you know that bacteria is 60% of your feces? You didn't, did you? Oh my God, that's a lot. Look at your bowel movement the next time and think of 60% of that. undigested material because we can't break down cellulose, things like that. They can digest some of that. They deter, believe it or not, they protect us by keeping harmful bacteria away because they compete with them. And then last thing, because the bacteria are them and we do see them as harmful, they will stimulate the immune system. And that's done through molt. And the big molt in the ileum, that's right before the large intestine, are the Peyer's patches. Okay. What are the two movements of motility of the large intestine? Again, segmentation or turning, it's a little differently. It's done by the circular muscle, similarly to the small intestine, and will swirl the water and electrolytes, allowing them to come into contact with the wall so they can be absorbed. And what's called mass movement. Without mass movement, how would you move anything through your colon when you're standing up? The first place feces has to go is from your cecum up. So basically, this kind of wave of contraction occurs to a length, forcing that amount forward. And that's called a mass movement. Two things will trigger a mass movement to occur. Your body's not stupid. You decide to eat, and it goes, oh my God, I still got stuff in here, I better get rid of this. And it's a reflex. It's called your gastrocolic reflex. Food in your stomach. Did you ever go, you just ate, and then you felt like, oh my God, I have to have a bowel movement? Just think about it. You don't have to nod yes or no. Okay, I don't need to know your personal bowel habits. But just think about that. Now you're going to think about every time you have a bowel movement, what's happening, aren't you? But that's good. And then maybe you get a question right on your thing. Also, food emptying your duodenum will do the same thing. Oh my God, there's something coming. We need to make room. Let's get rid of it. That's all this is about. All right, the defecation reflex. This was another active learning activity that we're going to do as an entire class. So, if no volunteers volunteer the answer, then we're going to move on. But anybody can yell out the answers, as long as it's not the same person every one, and they've already filled it out. Okay? People be quiet. What kind of receptors? Stretch, yeah, of course. Transmit the sensation of rectal distension to, anybody know the region? Yes, it is the sacral region of the spinal cord. It's way down here, just think where your anus is. What kind of neurons are we dealing with here? Parasympathetic. They cause what kind of muscle? Smooth. In the what colon? Sigmoid. And the what? The sigmoid colon and? Well, it's not your stomach. Yes, rectum, of course. And which anal sphincter will relax without you thinking about it? Internal, yes. Okay, impulses from the blank. Cerebral, or you can put cerebrum or cerebral cortex, that's correct. Trigger relaxation is what kind of muscle now? Skeletal. And now which anal sphincter? External. And then you will also contract your levator canine muscle. So, this will happen not with a lot of voluntary consciousness for you, but it just happens at the same time. But what you're going to do, because you're starting to feel it down there, you're sitting in A and B, and it's not time, you're going to voluntarily contract your external anal sphincter to what? The defecation reflex. Inhibit. Yes. You guys did awesome on that. So here's the figure. showing everything we just filled in. All right? Make sure you know which ones are the parasympathetic nerves, which ones are the stretch. So these would be sensory nerves here. And we know we're going to the lateral horn, and we're dealing with the autonomic nervous system. All right, what do you think?
I feel like last time to make this
I should have made everybody do this one on their own. Because it's really hard. All right, Trevor. What do we think? Oh, come on. You had to ask somebody what they put? If something moves fast through your colon, we call it what? Not, I know, you guys doubt you thought, is this for real? Yes. It's diarrhea. Don't put constipation. You guys know that that's not the same thing. All right. All right, there are all the notes. It's a wonderful thing for you to look at. There we go. Moving on. Pancreas, liver, and gallbladder. Well, he's not back, so there's a good chance it's still not being recorded, other than my voice. Okay. And I know that can be hard to follow. Or excuse me, follow. All right. First, the pancreas. It has both, so we're at the very top of the notes here, in that tiny little space. Endocrine and exocrine functions. I'm reminding you what the endocrine functions are. Insulin and glucagon, and of course they are released in relation to food. And of course the metastatin, which we've already heard about. The exocrine are the enzymes secreted by the acinar cells. Now, did you guys hear about those in lab? Yeah. So when you look at your pancreas slot, you saw the islets, which were the endocrine, and the acinar cells, which are the exocrine, making digestive hormones. All right, where is the pancreas located? It's nestled in the curve of the duodenum. That would be the head of it, and it's retroperitoneal, left upper quadrant, because it kind of starts here and moves over to the left. So you have a head, a body, a tail. I heard somebody was asked pancreatic duct on their practical. Anybody else asked that? You guys listening? Friday? People? Okay. I don't know. It's easy to ask. There's hardly anything else it could be. There it is, all nestled. So they're both retroperitoneal. All right, what are the two cell types? So now we are here at two. Pancreatic juice, similar to gastric juice, except now it contains water, digestive enzymes, and it's basic, as opposed to gastric juice, which was acidic. Two types of cells. So you have the ducts. They secrete the bicarbonate ions. And the whole purpose of a bicarbonate secretion from your pancreas is to neutralize the pH of 2 coming from your stomach. The acinar cells secrete the digestive enzymes that are going to break down, depending on what they are called, carbohydrates, proteins, lipids, and nucleic acids. We'll get to that at the very end of the lecture. I gotta move on. I'm sorry. So now we're at number 3. This slide fills in number 3. Let's see if you guys, when I have blanks, we can spread it out a little bit longer. Pancreatic secretion occurs to what kind of stimulation again? Parasympathetic still. By which nerve? Vagus. Still vagus nerve. And what two hormones? I've given you right here. CCK, cholecystokinin, and secretin. All right. What are the effects? Cholecystokinin will act on the acinar cells to secrete the digestive enzymes, whereas secretin acts on the duct cells to secrete bicarbonate. Those are the two things we want. They are both duodenal hormones that are released into the bloodstream and travel to your pancreas, where they'll have that effect. All right. And here is the figure showing the duodenum. There's the hepatic pancreatic ambulatory sphincter and the major duodenal patella. Enters. Here's the duodenum, secretin and cholecystokinin, secretin going to the duct cells for bicarbonate, and the CCK going to the acinar cells for the digestive enzymes. And together they make up the pancreatic juice. All right, the anatomy of the liver. I have to state the 4 lobes. My God, if you don't know by that, you haven't been even paying attention in lab. Let's move on. And there they are. Will you have a figure like this on your exam of the liver? No, but you have to know the lobes in case they show up in a question. All right, what is the flow of bile? So I've given you some notes here. And to discuss this, I have 4 terms. First, a liver lobule. You guys have already learned the first 2 in lab. The liver lobule is the functional unit of the liver, composed of hepatocytes, has a central vein in the middle, and at each of the 6 points, because it's a hexagonal structure, you have a portal triad at each. What does that contain? The hepatic arterial, the portal venial, and the bile duct. Then you have to know 2 circulations. The flow of blood into and out of the liver, and the flow of bile. The flow of blood begins with the hepatic artery, then to the hepatic arterial, or at the same time, because 2 circulations drain into your liver, the hepatic and hepatic portal. The hepatic portal vein to the portal venial, so we're at 2 of the triads. Then, to get to the central vein, we pass through the hepatic sinusoids. I'll show you this in a figure in a second. Then out all the central veins to the hepatic vein to the IVC, inferior vena cava. Bile goes in the opposite directions. Instead of going into the liver, bile has to come out. It's produced in the hepatocytes. Then you learned in lab the bile canaliculi. Anybody ask that on their practical? At least one. Okay, good. Then to the bile duct, then to the right and left hepatic ducts, then to the common hepatic duct, which will join with the cystic duct, and then to the common bile duct. All right, so that's the path. All right, I know you had to learn that for lab, also for those structures. All right, I'm sorry, I gotta move on, because we still have like 3 pages of notes, don't we? 3 or 2? Oh, just 2, okay. All right, just 2 whole pages, yeah. So everybody look here, see the arrows? This would have come from the hepatic artery to the hepatic arterial. This would have been from the hepatic portal vein to the portal venial. They air through the hepatic sinusoids to the central vein. The central vein will go to the hepatic vein and the inferior vena cava. For bile, you can see the arrows will come in the opposite direction. Hepatocytes make it to the bile canaliculi, which join the bile duct, then to the right and left hepatic ducts, then to the common hepatic duct, then to the cystic duct, combining it to form the common bile duct, which empties out the hepatopancreatic ampulla and sphincter. See, you'll be able to do that someday, honest. You'll be the smartest nurses, doctors, physical therapists, athletic trainers in the world. All right. If I left anybody else,
You know. What are you thinking?
I have no idea.
I'm thinking maybe a lot.
Like one maybe? What are you guys thinking? Mitochondria.
I mean, it does all this thing.
Don't take my word. I feel like every time you take my word off, you're gonna get it
Gonna be
off.
all of them. I'm gonna go back to my all of them.
I'm just gonna go to all.
All learn. There's a lot of the liver. There's a lot of. I know, man. I can read this. And we all in the middle. I even understand in like 3 days. I told my parents all the time, I'm like, you need to just sit in one of these classes and you'll understand the pain I go through every day. I'm like, not just the Tuesday, Thursday. I literally, I'm be like,
and if we have time for that, you'll get the points. All right. When you get up the PRS question. Ta-da.
hey.
All right. All of them are found in large quantities. Why? Well, if you're looking at the major functions of the liver, and if they're a major function, that means whatever doesn't has to be found in large quantities. Okay. I gotta quickly go through this. This was a fun, active learning for us. You guys have the beginning of the notes, and now we're gonna match more specifics with it. You ready? Here we go. All right, which function goes with stores fats? It's called lipid metabolism. Okay. Which one goes with A, B12, D, E, and K? Stores vitamins. Don't be stupid with some of these. You'll be what regulates blood levels of lipids? Lipid metabolism. This is where cholesterol, HDL, LDL, and so forth. And of course, triglycerides. Which one would break down amino acids? Which function does that go to? Anybody know? Protein metabolism, of course. C. What about sugar, glycogen, glucose? A. Carbohydrates, right. Sugar, glycogen, glucose. That's all you got time to write right now. Or just stare at me and write nothing. That's fine. Blood clotting what? What are they called? What did we learn? They're called clotting factors. And what are they? They're not vitamins. Proteins, they are. So they go with protein metabolism. What's albumin? It's a plasma protein. That goes with protein too. You guys are doing well. I can see most of you are getting this down if you're not trying to do word for word. That also produces enzymes. Those are proteins. But what are those enzymes for? Detoxifying chemicals. So that goes for D, removal of drugs and hormones. Hepatic macrophages phagocytize. Oh my God! H, phagocytosis. Why? What's that special name for those cells? Anybody remember what the hepatic macrophage starts with a K? Kupfer. K-U-F-F-P-E-R. No, K-U-P-P-F-E-R. Kupfer. Okay. They phagocytize just like the spleen. Damage red blood cells, so that goes up up to excretion of bilirubin, because the breakdown of red blood cells leaves us with bilirubin, and the liver excretes that. They also store iron, so iron stores would go with G, storing minerals. And which one would emulsify fats go with? Bile salts, because that's the big word. Okay, this is on your exam. You guys, it looks like most of you got time to do it. All right, now we got to move on to another PRS. Here we go. Also, make sure you understand the organelles,
Yeah.
like in these PRS questions, please.
I agree.
It all comes back.
I agree with that assessment. I agree with that assessment.
Why? Wait, why?
Polypeptides make up proteins. Smoothie doesn't have anything to do with proteins. That's just lipids and drugs.
Yeah.
No problem. Anytime.
Good
man.
explanations.
I know. One question I actually understand, but you know what? The answer will probably not be one. Just because.
Just for one person. The last one, I was like, one.
Oh, that was smart. Haley. Hey. I was making faces at you, and you're like looking over here like, and you're like, I'm like, Haley. I have a wedgie. Sounds good.
Alright. I thought you guys needed a breather. You got an extra second on this PRS.
Oh, wow.
Do you have a hand cramp yet? Close. Alright, which is not a function of the smoothie R because it's a function of the roughy R. It's not 5. Did you guys get it right?
Wow.
Polypeptides are what? Proteins, and proteins are the roughy R. Everybody got that? Okay.
That was easy.
Not done yet. You do get lots of PRS points today. Just rapidly.
I know you can't eat nuts because my mom has problems with their gallbladder, and she's like, "Hey, nuts."
I feel like there's fat in milk.
Huh?
I was thinking it was fat, but I'm not sure. But I feel like fat's like a generalized.
But when you break down,
Bile salt and emulsifies fat.
okay.
So you break down in it.
All right, what do we think?
Not salt.
I hope it's not salt.
I was thinking protein or fat.
Is it going to be 100? No.
Oh, okay, got it right there.
I was like, maybe I should.
Yeah, because your gallbladder, just watch your salt. No, that's for hypertension. We've learned about that. But the gallbladder stores bile, and bile helps emulsify fat.
Yes, smart.
Yeah, they get what's called a Dunkin' syndrome.
Did you get that one?
If they go and then
That was
like
Maddie.
KFC,
Why didn't you share?
because there's not
The one that
enough
Maddie
bile
knows.
coming- that's not
What? This is the one you know.
It's called steatopia, and it floats and stinks because it's fat that never got digested. Can you imagine something like oil going through your gut? It's going to slide right through very quickly. Nothing's there to break it down. All right, what about the gallbladder? And unfortunately, you already know this. You know all the duck names and everything. But its whole point is to store bile, and then under the influence of CCK, it'll contract it, and I'll be getting to that, with the state to control of formation and secretion of bile. All right, I gotta move on. The gallbladder is not rocket science. It's pretty much does one thing. Any questions on gallbladder? What kind of muscle is it? It's still smooth. All right, there it is, all cute. There it is. Look at those stones. They look like a string of, it's like somebody ate a string of pearls and it went in their gallbladder. They don't look that good. But yes. All right, this is more important. Make sure you get this one in here. So I'll try to slow down a little bit because there's not a lot on here, but this fills in state of control. First of all, the liver produces bile continuously, all the time. But it only secretes it in response to the gallbladder. If the gallbladder contracts, then more is going to be released by the liver because it's going to have to fill it back up. It will also relax the hepatopancreatic ampulla and sphincter. That makes sense because we want to get that bile in the duodenum. The hormone for the relaxation of the ampulla and the contraction of the gallbladder is CCK. Cholecysto is the combining form meaning gallbladder. Anytime you see that. Choledocho is the common form meaning bile duct. Secretin will stimulate the production and release by the hepatocyte. So secretin acts on the liver, CCK acts on the gallbladder. What happens though is bile salts, after they go into your gut, will re-enter your circulation and go back to the liver and have a positive feedback loop because I told you that bile, the liver makes bile, yes, continuously, and that's part of that loop. All right, here's the figure. Love this figure. Here's the gallbladder, here's the liver, here's the duodenum. Duodenum, after chyme has entered, secretes secretin and cholecystokinin. We already learned that they have two effects on the pancreas. They also have two effects, CCK on the gallbladder, secretin on the liver, and bile salts also back positive feedback loop on the liver. All right, any questions on this? It's clear as mud. There's just as much on your test on digestive. I apologize for having to go through it so fast.
Okay.
Boy, memorize that one. Okay. Actually, I think you're going to have a little bit of time. Here we go. All right.
What's going on?
Nutrient digestion and absorption.
We need pollen. To maybe make this some paper right there. I'm like, okay, just.
All right. So we've broken stuff down. First, the
You
overview.
don't like hit stop and say, no, stores.
What types of digestion do we have? Which one physically breaks it? Yes. And which one chemically breaks it? Chemical. It's not a trick question. Yes. Most digestive enzymes catalyze what kind of reactions if it's digestive? Hydrolysis or hydrolytic, they mean the same thing. Yes. Awesome. So what do they use? What kind of molecule? Water, of course, because that's why it's called hydrolysis. To break the bond, what speeds it along? Digestive enzymes, yes. All right. Helping you out. How about a PRS? Always give you some points as a PRS.
Three. What am I doing? Sorry. But is embryonic the same as hydrolysis? Is extradonic? Oh, wait. I'm just going to stick with hydrolysis because I don't want to, like, yeah. If I just get one right, I get one right. So that is.
Wait, what did you put?
Oh man.
Whether you think so or not. Okay, what's the answer?
It's not.
Of course, it's still hydrolysis, but what could I have said? Exergonic, yeah, right? Do you know the number one way to make yourself warm is to eat? The amount of heat you generate by breaking down all those bonds in your food, you'll get warm. So, eat on a cold day. All right. There it is, hydrolysis reaction, blah, blah, blah. Okay, next one. What do you think? Oh my, we still got a ton to do, don't we? 10 minutes.
Wait, yeah, because this is simple.
Lipids.
So make sure
Take
that
it up.
there's nothing wrong with those here that you have just like most of them, yes. So,
I know it's not one because lipids are taken up by, like you said, some dietary fats is like.
Yes. So is it 2 or 3?
No, that's not. We know it's not lipids, so we're saying 5.
all right, let's work on that.
Because if you make hemoglobin come,
Yeah, the only thing I was trying to remember was monosaccharides. Something to
Hurry,
carbohydrate.
Okay. Well, it's probably not lipids. Why? That's right, they can pass through. So I know that, and I know you guys know that because I taught it to you, so I would have picked, and you did too. Yeah. In the gist, this is it. Lipids, simple diffusion is going to come into play. It's not as simple as that, but it's part of it.
I think that's right.
Facilitated diffusion is how we bring in by, anybody know?
I'm sure it's one of them pancreatic juice for breakfast.
Well, tell you in 1 second. Here we go. So, there it is. It comes in one side. I hope you guys figured it out. And by transcytosis comes out the other end. Blah, that's it. Okay. So let's digest and absorb carbohydrates. Did I give you most of these notes?
No.
They're broken down by salivary what? And pancreatic what? What goes in both blanks? Amylase, of course. And then we have 3 brush border enzymes. Remember I told you they break down the disaccharides? And the 3 are maltose, lactose, sucrose. So that's why they have those 3 enzymes. Glucose and galactose, secondary active transport. You guys remember that way back from AP1? And then into the blood by facilitated diffusion, as you just told me. Fructose does not need ATP. That's why what is bad for you? And you absorb it really fast, and it screws with your sugar levels in your body. Fructose only has to come in passively. That's why in high fructose corn syrup, they're finding out it's not the same thing as other sugar, which has to be broken down. All right, here's the figure for that. Here's fructose coming right through by facilitated diffusion, secondary active transport, followed by facilitated diffusion into the blood. Absorption of proteins. Proteins first have to be broken down by amino acids in the stomach, and we know that enzyme there is pepsin. I'm really out of my mouth. It's coming out the gist of what I want you to know. So, but most protein digestion takes place in the small intestine. By the pancreatic enzymes that I have listed here, and the brush border enzymes, which I've listed wrong here. Those are there, but they're not breaking down proteins. I apologize for that. They're called peptidases. What do you think dipeptidase would break down? not a disaccharide, dipeptide, yeah. I apologize, that shouldn't be there, the maltase, lactase, and sucrose. Okay, how do we digest, or absorb proteins? Again, we have secondary active transport, active breakdown, and then facilitated diffusion into the capillaries. All right, digestion and absorption of lipids. Two methods. Believe it or not, a big part is it just physically tries to break the bonds. And it's mechanical. Mouth, stomach, intestine. Then we have to emulsify it with bile salts, because bile is an amphiphilic, just like phospholipids. It has a polar and a non-polar end to try to break them apart. That's called emulsification. All right, what are the 4 steps of lipid digestion? Excuse me. Yep, and lipid absorption. Wow, still got a lot. Okay, gastric lipase. Lipids enter the small intestine and are emulsified is the next leg. Pancreatic lipase catalyzes, and the bile salts form structures called micelles that look like little circles that surround a fat globule. Okay, everybody got these 4 blanks? All right. Describe the process of lipid absorption. We're at 3. Here, micelles, get lipids across the plasma membrane. Then they enter by passive diffusion. Believe it or not, they're reassembled into triglycerides. So we break them down, then put them back into triglycerides because it's the #1 storage form of lipid in our body. And then they circulate through chylomicrons, which are much, much larger than HDL and LDL. And we know they have to enter a what? Because they're lipids. A lactate. All right, briefly discuss absorption. Guess what the enzymes are called that break down nucleic acids? You can't guess? They're called nucleases. They still end with ACE. But what I want you to get out of this is what do we break down the nucleic acids into? The phosphate group, the sugar, and the nitrogenous base. Okay? So phosphate, whether it's DNA or RNA, it'll be ribose or deoxyribose, and nitrogenous base. And it is absorbed through primary and secondary active transport. All right, well, we got one more thing. Briefly. We do. We only have one more thing, don't we? Absorption of water, electrolytes, and vitamins. I hope they get this fixed by next Thursday. Don't you? How many of you really listen to the Echo 360s? Wow. Okay. Really? Because when I look at the numbers, it's never that high. No. Yeah, blame everything on them. All right, here's our last slide. We absorb, of course, water, electrolytes, and vitamins. And did you know most of your, even though the large intestine, one of its main functions is water, the majority is absorbed way before that. And, of course, water is through osmosis. familiar fat-soluble vitamins are lipid-based and interlacteals. A lot of you didn't go, I put fats and water-soluble vitamins.